Okay, thanks very much to the organizers for allowing me to give this talk. I didn't realize there were so many people in the room, so it just turned around. So, I and a few colleagues have been trying to develop this field we call evolutionary cell biology. I'm very interested to see if this is exactly the same as evolutionary systems biology. I think there's a lot of overlaps. What I like about this area, both of these areas, is this, that we have this odd situation in evolutionary biology. We like to do comparative biology and just make up a story about adaptation. You're doing evolutionary biology. But we really need to understand evolution. We really need a deeper mechanistic understanding of what's going on. And this means bringing together the confluence of at least 3 different areas. There's a lot of good people from physics now moving into biology, really doing their homework. And we aren't allowed to violate the laws of physics, so we have to pay attention to what's going on in here. This field's almost as sophisticated as the one in the middle, population genetics. No population genetics, no evolution. And then finally, biochemistry, of course, tells us the laws of, that we must obey in molecular biology. So I'm viewing this whole area as a confluence of 3 of our big areas in biology that have a good theoretical basis. Genes we're all made up of genes, and everything that comes from genes. Genes do very many wonderful things for all of us. But there's also some baseline cost of having a gene or even having a fragment of DNA in a cell that's doing nothing at all. There's a couple of costs that we need to worry about. And it's mainly the costs that I'll be talking about today of a gene, because these are the baseline currencies that you have to pay before you get any boost from any product of the gene. So one of these costs I'll give some brief overview on is what I would call a mutational cost. All excess DNA is dangerous stuff to have lying around, and any embellishment you want to add to a gene increases the mutation rate to defective alleles. So we have to worry about that. I'll give you an example in a second. And then there's a bioenergetic cost to a gene at many levels. These costs come in at the DNA level, the RNA level, and at the protein level, as we'll see, in terms of the energy that's used to maintain and run a gene, and we use currency in terms of ATPs. And what we mostly are interested in here is how expensive are both these levels of the cost relative to the power of random genetic drift? Because if they are higher than the prevailing role of random genetic drift, then selection can easily modify things. And if they are not, they will just simply accumulate by mutation pressure in genomes. You could probably add other costs to this list here, but these are the 2 I'll talk about today. Let's see. So I've been sort of informed that not everybody knows what drift is, and it's sort of a mystifying force. And so I want to very quickly here demystify random genetic drift. All populations are finite. And so if we imagine a tiny population here with a certain allele frequency, if you follow it, there's a population there's a gamete storm produced by that population. We come up with 10 individuals again, and so on and so on. And you see that over time, that allele frequencies fluctuate from time to time, and eventually we'll end up with one flavor of allele versus the other. This process scales with the population size. So it doesn't matter what the population size is, there'll still be fluctuations in allele frequency from generation to generation. But if the population is, say, 10 times larger, then it takes 10 times as long to get to the same place. And what's important in evolutionary biology is the magnitude of these fluctuations. If the fluctuations in allele frequency just due to drift alone are greater than what you would expect from the deterministic force of selection, drift wins. So that's really mainly what you need to know. And you should keep in mind, too, that drift is largely a random process. So if you start at the same point in replicate populations, you'll go in different directions and eventually fix with 50-50 probability each allele if you're starting at a certain point. Now, some populations are really, really large, and you might think, well, random genetic drift doesn't apply to microbial populations. Well, maybe it doesn't in the terms of the numbers of individuals, but there's still important levels of stochasticity in things with very large population sizes, such as microbes. And that's due to the unfortunate fact that all our genes are typically glued together on chromosomes and therefore inherited together, and that causes selective interference. So imagine we have a situation here where you have one advantageous mutation. These are just a small sample of chromosomes. Little tick marks are background polymorphisms, and this is a good mutation, and eventually it's pulled to selection, and in doing so, the segment around that allele has not had time to recombine, and that tick mark goes to fixation as well, but all the other variation of the population is lost. And that can cause a couple of problems. For example, one of those tick marks, this red ball here, might have been another advantageous mutation, but not quite as advantageous as the blue ball there. And so it's actually lost due to selection, due to the fact that linkage is going on here. And the other thing that might happen is this guy might have been linked to this little green polymorphism there. That's a deleterious mutation that got dragged along to fixation. Now, if we had free recombination, none of this stuff would happen, and we would have ended up with fixation of the red ball, one beneficial. We wouldn't get the green ball, no fixation of a deleterious mutation, and we'd get this blue ball. So basically what linkage does is it reduces the efficiency of selection. And even in very, very large populations, this puts a damper on the global effective population size. Here's some numbers just to give you an idea of what effective population sizes are. I won't go through the way we get these, but they come, as many of you know, they come from neutral measures of neutral polymorphisms. This is actually the absolute numbers of, a plot of the absolute numbers of individuals versus the size of individuals. A log-log plot with a slope of -1. And so this implies that every species roughly commands the same amount of biomass on the planet. There's 20 logs here. Now, the effective population sizes do not vary by 20, and this is due to the fact that once the numbers get very, very large in a population, it's not numbers that matter anymore, it's linkage that's mattering. And so if you look at big clunky things like trees and vertebrates, effective population sizes are down around the order of 10 to the 4th, 10 to the 5th individuals, often much smaller than the absolute number that ecologists would count. But even microbes, the effective size is typically no greater than 10 to the 9th individuals. And I guess a pile of 10 to the 9th E. coli cells you'd barely be able to see. So this is probably the upper limit to effective population size in any organism. Let's be generous and say 10 to the 10th. And so what this means is that in big multicellular organisms, the power of drift is equal to the reciprocal of the effective population size. And if that's greater than the magnitude of selection, then drift wins. So what this is saying is that most deleterious mutations with selective coefficients against them smaller than 10 to the -5th can't be seen by the eyes of natural selection. Whereas in bacterial populations with effective size on the order of 10 to the 9th, changes that reduce fitness by only 10 to the -9, a very, very tiny change that you'd never be able to measure in the lab, can nonetheless be seen by natural selection in the field. So let's think briefly about this cost of a gene at the mutational level. And this is sort of a summary of a lot of work that I've and a few others have done over the years. And so on the left, I've plotted out sort of in cartoon fashion the extremes of genome architecture. So here we have a typical segment of a prokaryotic genome. So these are like the Priuses of the biological world, nice, streamlined, elegant genes, no intervening introns, very small intergenic DNA, usually only 1 or 2 and sometimes 0 transcription factor binding sites. And then we get into eukaryotes, big, messy genomes, the epitome of unintelligent design. We have our genes split by intronic sequences. Those introns have to be transcribed and then spliced out subsequently. So in things like humans, these exons are only about 150. The coding exons are only about 150 bases on average. They live in this gigantic sea of intronic DNA. We have big upstream and downstream trailing regions on transcripts called UTRs and often very complex regulatory regions. All of these embellishments that have been added to eukaryotic genes can be viewed as a dangerous thing to have. For example, a upstream UTR can be dangerous because it provides a platform for a mutation to a premature start codon. And if you start in the wrong start translation in the wrong place, then 2/3 of the time you'll be out of frame and you'll have a defective allelic product. Introns, of course, are a problem because if you're going to have an intron, you have to splice that intron out at exactly a precise place, otherwise you'll have a downstream frame shift. And even DNA that we might all agree is completely innocuous, non-functional innocuous, completely non-functional DNA can be dangerous stuff to have. If it's non-functional, obviously you can't suffer from a loss of function mutation, but you can suffer from a gain of function mutation. It can cause an adjacent gene to turn on in the wrong time, for example, during development. And what we see is this is the scaling of various aspects of genome architecture with genome size. And what we see in small things, almost all the genome is coding DNA, and then eventually you get to genomes that are about 10 million megabases or up to 100, and then things level off, and most of the expansion in DNA in a genome is now due to intronic and intergenic DNA. It's almost a threshold sort of behavior here. And so we think this is due to the fact that most embellishments in eukaryotic genes are deleterious, but they're very, very mildly deleterious, and I'll give you an example in a second. So they can be easily selected out here, where effective population sizes are really large, but not here. They will just passively accumulate over time. Let's think about this with respect to a common kind of embellishment in eukaryotic genomes. There's about 100,000 or so introns in the genes in the human genome and are coding DNA. And we can make an estimate of how dangerous these things are from a mutational perspective in the following way. So what I've done here is summarize data from about 30 or 40 human genetic disorders. So these are monogenic human genetic disorders. And they're very debilitating disorders, and they're dominant. So mom and dad don't have the mutation, and the phenotype is very obvious. Kid has the mutation. You sequence the genes in the kid and in the mom, and you can find out the causal mutation. And we know what the genes are for all these things, and so you can go to any one of these genes, and you can count the introns in the gene. You can count the number of coding nucleotides, as we do in any gene. And then you can ask the question, if we have several hundred de novo mutations that cause the disease, we can ask how many of these are causing the disease because of a splicing problem, and how many are causing the disease due to a missense problem, an amino acid change in the coding region. And it turns out the ratio of this to that gives you an estimate of the number. You can think of a nucleotide as being equivalent to a certain number of key nucleotides in the coding region. That would be its danger. And these, you can see all the data here, and that line there is 30. And what that's saying is basically adding an intron to a gene mutationally would be like adding 30 nucleotides that you wouldn't be able to touch, otherwise you would have the disorder. So that's sort of the price we pay for having an intron. It's like adding 30 critical nucleotides, so the mutational cost would be 30 times the mutation rate per base. That mutation rate per base is 10 to the minus 8 in humans, so the penalty of an intron would be about 3 times 10 to the minus 7. So the human effective population size is somewhere on the order of 10 to the 4th, so there's no way in a population genetic context like humans you could prevent the proliferation of an intron based on its mutational danger, but you could easily do this in a microbial species. Just to give you an idea, about 8% of us are going to die due to some change in an intron in some cell in our body. That's a back of the envelope calculation. So, okay, now, so that's sort of the mutational issue. And what I really want to spend my time talking about today are the bioenergetic cost of a gene. How much does it cost to run a gene at all its levels of manifestation in the cell? And I've done this work with this guy here, Georgy Marinov. He was heavily involved in a number of the ENCODE projects. He's now in detox in my lab as a consequence of that. We've been working on a number of issues here with respect to the cost of a gene. And I'm just outlining a few of them in words. But the main point here to think about is there's 3 levels of cost of a gene. There's levels that you have to pay at the chromosome level. You have to make nucleotides. You have to replicate those nucleotides. And the cost becomes much higher in eukaryotes because you have to wrap those chromosomal segments around nucleosomes, so you have to make the histone proteins and so on. There's cost of polymerization to chain link the nucleotides together and so on. There's costs at the transcription level, and those costs can depend on the rate at which you transcribe a gene and also the rate at which you decay transcripts. Again, there's a price to be paid for ribonucleotides that have to be made. There's a bigger price if you have high turnover because you have to constantly replace the transcripts, polymerization costs. There's bioenergetic cost displacing, and the list goes on and on. I'm not going to go through all the details, but the numbers that I'll be giving you are all based on all these different levels of cost. And then finally, there's a cost at the level of protein, We have to build amino acids. We have to, again, tape those together to make our proteins. We have to worry about the turnover rates of proteins and so on. Most, as we'll see, most transcripts and proteins have very short half-lives relative to the lifespan of the cell, so you have to continuously produce these. Now we measure, from an evolutionary perspective, we measure the cost of a gene by adding all of these things together and dividing by the total cost of building a cell. The way to think about this, there might be a few grad students in here, and I forgot to check the currency, but I would guess that if you paid $20 or let's say, okay, pounds, you use pounds here, right? So if you're a grad student and you spent 100 pounds on dinner, I guess you would be feeling some real pain if you were a grad student, right? But there's this guy, Donald Trump, that you've probably all heard of. You could pay 10,000 pounds and you wouldn't feel a thing. So that's the sort of the point here. So we have to scale everything by the cost of a whole cell. And we use as a universal currency, we use ATP hydrolyses. So the way to think about this, the cost of a gene is the sum of these three different levels of cost. And the net advantage of a gene, genes can make a product that do other wonderful things for cells, improve our survivorship, improve our reproductive capacity, and so on. That would be this here. And the net advantage of a gene is the sort of the advantages you get as an ecologist would think about it, subtracting out the bioenergetic cost. And again, the key thing with respect to drift and selection is we want to know whether these costs are smaller than the power of random genetic drift. If they are, they're sort of irrelevant with respect to selection. But if they're not, if these costs are greater than the power of drift, then selection will keep various modifications of genes out of populations. So to move forward here, the first thing we had to do was we had to figure out, well, just how much does it cost to build a cell? And it turns out there's a fairly simple way you can do this if you know a bit about metabolism and you can grow your bugs in chemostats. So many of you know about chemostats. These are closed cultures. You have sterile medium here going in and you have a reservoir going out, and they better both be flowing at the same rate or you'll have a problem with your chemostat. And the way this works, your bugs are growing in here in this sterile media. They're being fed from here and consuming resources, and they reach a steady state density, and the growth rate of the cell has to equal the flow rate of the chemostat. And so what you can do then is you can grow your bugs at various different flow rates, different dilution rates. Those are different growth rates. And you know the amount of a nutrient here. You usually have a carbon source that's both the carbon and the energy source. You know the amount in there, you know the amount coming out, so you know the flow rate, so you know how much is being consumed, you know the number of cells in there, so you know the consumption rate of the resource per cell per unit time, and then you can plot this as a function of the growth rate of the cells. And then if you know some metabolism in your bugs, you can convert the consumption rate, say, of glucose into the number of ATPs that are produced. Now the intercept there has a simple interpretation. That's the amount of ATP the cell is turning over per unit time when it's not dividing. So that's the baseline maintenance cost. The slope of this line then gives you a measure of the amount of ATP required to build just to build the infrastructure for a new cell. So I'll be talking about both these levels of cost. One's per unit time, the maintenance, and the other is just the amount of total ATP to build a new baby cell. And I've summarized data here for, I don't know, about 20 or 30 different species. There's three colors here. These plots are all with respect to cell volume. So what we're inquiring here is how does the cost of building a cell scale with the volume of the cell? And you can see that these are log-log plots, so-called allometric relationships. And you can see the exponents here are neither one significantly different from one. So this is isometric scaling. The cost of building a cell scales with the volume of the cell. So we can say things are isometric. I'll point out that all these black points here, these are all for bacterial species. The blue points are all for unicellular eukaryotes. The red points are for multicellular eukaryotic cells having an out-of-body experience. So they're cell cultures from multicellular organisms. These are not actually used in this regression here because of a worry that cells don't necessarily behave like they should when they're growing in culture taken from a muscle or something. But you can see nevertheless they fit quite well. One point that I'll return to, there's no break. There's complete continuity of scaling in the cost of building a cell across prokaryotes and eukaryotes, as though we're nothing more than the extension of what you see metabolically in a eukaryotic cell. And then the lower level plot there is for the metabolic cost, the maintenance per hour requirement. Just to give you give us a benchmark here. A 1 micron cell is roughly equal to an E. coli cell, and it costs about 27 billion ATP hydrolyses to make a baby E. coli cell. Because of the isometric scaling, it'd be about 100 times higher for a yeast cell, and about 1,000 to 10,000 times higher for a typical vertebrate cell with this linear scaling. So again, I've already pointed out that there's continuous scaling between, there's no dichotomous break, so nothing's suddenly excited to happen metabolically when we moved into the eukaryotic domain. And I'll just leave this as an open question for people in evolutionary systems biology. We really have no idea exactly why these slopes are one, and we certainly don't have any idea why the intercepts reside where they are. So this is probably something that should be tackled by a good biochemist and good biophysicist, but we really don't know. You might call this a scaling law, but we don't really know why that law exists in terms of the exact positions of the slope and the intercept. Now, this is a different kind of plot here, again, versus cell volume. It's about 11 orders of magnitude cell volume. These are bacterial cells, and these are various other eukaryotic lineages. This is the minimum cell division time. I'm putting this up here to make a point. I just showed that the total amount of ATP required to build a cell scales with cell volume, but that doesn't have a time dimension in it, of course. It turns out that many of you will already know this. These are minimum cell division times for various bugs, and many of these are based on lots of observations in lots of labs. There's a lot of noise here, but each point's a different species. And what you can see in general in eukaryotic lineages, the bigger the cell, the longer it takes to divide. So it's taking more ATP to make a baby cell and also more time to make that cell. So the energetic efficiency is going down. On the other hand, for bacteria, there's actually a somewhat negative slope between the cell division rate and time. We don't have a whole lot of room to work with bacteria here in terms of range of cell volumes, but it's certainly not a positive relationship. And so what this ends up telling us is that bacterial growth rates on a per unit volume basis, larger bacteria are actually incorporating energy into biomass at a higher rate per unit time as they get bigger. And you have the opposite sort of scaling going on in multicellular, in eukaryotic organisms. So completely different scaling with respect to bioenergetic efficiency, which I'll return to in a few minutes in the context of a broader hypothesis about the evolution of eukaryotes. This line down here can be viewed as the growth rate speed limit for cells. And we have no idea why that line resides exactly where it is, but we have a lot of data now. And what this is telling us is nobody in nature has figured out a way to have higher, despite the fact, presumably, of selection favoring higher growth rates, nobody is allowed to break that speed limit. So again, we don't know why that form of relationship exists, but there you go. Okay, so now let's try and think about how we would calculate the cost of a gene. And very little work has been done on this in the past. One person in the room has worked on this a bit, Andreas, with some earlier work, and some of you may be familiar with Hiroshi Akashi's work from many years ago. But now we have much more data to bear on these issues. So the way to think about this is you've got an organism consuming some kind of carbon energy substrate, starts to go through glycolysis or something, and you're starting to pile up various precursor molecules. And we use the precursor molecules to make the building blocks in cells, nucleotides, amino acids, and lipids. So the way to think about this cost is that you're starting with this precursor molecule, you're making some building block down here, and in doing so, you're consuming ATPs. So that's a direct expenditure to build a building block for a cell. But from an evolutionary perspective, there's another cost to worry about, and that's the indirect loss. Because if you use this precursor molecule to run your gene or to make DNA, make protein, and so on, that precursor molecule is no longer available to be metabolized and to produce ATP for other uses in the cell. So that's an indirect consequence of using up a precursor molecule. And the sum of these two are the total cost that you would have to think about from the context of from an evolutionary situation, because this means you can't do something else if you want to do this. And there's various ways to go about this. You have to do a lot of slogging through biochemistry textbooks and so on, but roughly speaking, these are the cost of building. This is the average cost of building an amino acid. The average amino acid is about 27 ATP hydrolyses. Nucleotides are a bit more expensive. Lipids that I'll return to later are quite expensive to make. Now, one other scaling relationship that I'll point out here that enables us to estimate the whole distribution of the cost of all genes in a few bugs comes from this. We have a few organisms now that we have good single-cell biology. We have censuses of the numbers of proteins and number of mRNAs in cells. In fact, the whole distribution in many cases. And this is just a summary of that data. This is for protein molecules. Again, we're looking at about 6 orders of magnitude of different cell volumes. So mammals are up here. There's bacteria down here. And I guess the 2 yeast are these guys here. And you can see, again, there's this allometric scaling. So across a complete continuity of scaling between bacteria and eukaryotes, and that slope there is less than 1. So cells, as they get bigger and bigger, are getting more and more dilute, actually, with respect to proteins. These are the mean number and the mode number of proteins in a cell, averaged over all the proteins. And the lower panel here are the comparable data for mRNAs. These are just distributions at various points of time. These things typically have log normal distributions. I want to point out here, this whole gray area here is, that's a value of 1. So many genes in really small prokaryotes have an average number of transcripts less than 1. So many cells are devoid temporarily of transcripts. And this is the kind of thing we have to worry about when we're thinking about the overall cost of a gene. So I've just given you a little background. Now I just want to summarize some data. Let's just take a look at the upper left panel first. So this is the full distribution of the 3 costs of all the genes in E. coli derived from a lot of biochemistry, a lot of genomics, and a lot of proteomics and transcriptomics. The details I won't go into, but I pointed out the basic fundamentals here. So just to give you an idea, the lower scale on these plots here, that's the number of ATPs required on a log scale. So the red line there, that's the distribution of DNA level costs for the genes in E. coli, and it's a 5, so the average gene at the DNA level costs about 10 to the 5th ATPs. The gold sort of distribution there is the cost of transcripts. This can actually be less for many genes in E. coli, again, because to replicate DNA, you have to replicate 2 strands, but if you have only sort of 1 transcript in the cell per generation, you're actually paying less for transcription. And then the yellow line there, which eventually coincides with the blue line, the yellow line's the cost at the protein level, and the blue line's the sum of all these, the distribution. So that's the total distribution of the cost of all genes in E. coli. Now shift to the top panel. All I've done in that top panel is divide the total ATP level cost by the total number of ATPs it takes to build an E. coli cell. So this is our measure of the evolutionary cost of the gene. How much ATP are you using to run that gene compared to the cell's whole energy budget? These lines over here measure the power of random genetic drift in E. coli. E. coli has an effective size somewhere in the neighborhood of 10 to the 9th, and I've padded it by an order of magnitude in both directions, just to give you a rough idea where we are. So this is our benchmark in trying to understand whether the energetic cost of a gene are something that can even be felt by the cell, by natural selection operating on E. coli. And so the benchmark here is 10 to the 9th, and if a cost is to the left of that, it's too small to be seen by the eyes of natural selection. And if it's to the right of that, the energetic cost can certainly be seen by natural selection. So if you had a piece of DNA over here, and you can see the DNA level cost over here, they're about 1,000 or almost 10,000-fold higher than this benchmark. This means that even a completely inert piece of DNA can be, just the cost of having that in your cell and having to replicate it, not even have to express it, is enough to be felt by natural selection. So this means selection can effectively see an insertion of just 1 or 2, certainly 10 nucleotides in a bacterial species and eliminate it if it's a mutant allele. So this helps explain why many bacterial genomes are very, very highly streamlined. It can be explained on a bioenergetic basis. Now we move across these other panels here. So here's the yeast, and there's the worm, and there's the plant. And what you may notice is that the absolute values of these distributions are moving to the right. The absolute cost of genes do increase for a variety of reasons. More transcripts are made, longer coding regions, things displaced, and so on. These absolute values are going up as we move to more and more complex organisms. But you'll notice what's also happened, the position of these bars here. The effective population size is going down dramatically as we go into yeast, to worms, and to Arabidopsis. And so now these peaks are no longer to the right of the power of random genetic drift, they're to the left. So what this is saying is, for example, Arabidopsis, you could put a segment of DNA up to 100,000 bases or so, not expressed, so it's not making a toxic product. But the bioenergetic cost would be small enough that Arabidopsis would never feel. It could never be eliminated by natural selection. So to summarize, what this is telling us that for bacteria, the cost of insertion of things as small as just a few bases can be felt by selection, the energetic cost. Whereas when we get into multicellular species, really large insertions, even if not expressed, can't be felt by the eyes of natural selection. Now the next panel summarizes a lot of other data in a whole variety of eukaryotic microbes here and bacterial species, again, plotted as a function of a cell volume. There's the 3 different levels of cost: replication, transcription, translation, and the total. Sort of a rule of thumb here is about 90% of the cost of a gene, if it's expressed, is typically at the protein level. About 10% is at the RNA level, and about 1% is at the DNA level. But the main point I want to make here is that there's a negative scaling of the cost of a gene. Again, keep in mind that this is relative to the energy budget of a cell. So what this is telling us is things that have larger and larger cells, going from small bacterial cells here to larger cells from multicellular eukaryotes, the cost of adding a gene, just the basic bioenergetic cost, is actually going down as the cell gets larger. It makes a smaller and smaller contribution to the cell's total energy budget. So this means it's easier for a large segment of DNA, expressed or not, just to passively go to fixation in a population of multicellular organisms, where it's very, very difficult to do this in microbes. Okay, so now I want to take a slight shift here because I've given some results that bear on a hypothesis that was recently suggested in this paper written by Nick Lane and Bill Martin. Hopefully they're not in the room. I'm sure Bill is, and he'd be screaming by now if you know Bill. So they published this interesting paper a few years ago. This is a line that comes out of the abstract. And the basic point of this paper is, the basic idea is that without the mitochondrion, there would never be any eukaryotes or anything that we associate with eukaryotes. There are many novel protein folds that wouldn't exist. There'd never be any expansion in gene number because genes are very expensive, especially in eukaryotes, although I've just shown you that the cost of a gene actually declines as you get into larger and larger cells. There'd be no internal complexity of cells, no internal membranes, there'd be no multicellularity, no development, no complex behavior, not even any sex. So they argue that the mitochondrion as the powerhouse of the eukaryotic cell provided a bioenergetic revolution that enabled us to expand our number of genes by 200,000. I don't know where the number 200,000 came from, but you know, E. coli's got 5,000 protein coding genes, so you can do the math. Maybe that was just a typo or a sticky 0 or something, I don't know. But anyhow, you get the basic idea, and it's sort of an enticing idea. We think of the mitochondrion as the big, you know, it is the powerhouse of the eukaryotic cell. So we want to evaluate this in a little more detail. I've already shown you some data that are quite incompatible with this hypothesis, but just to back up, for those of you, I'm not a biochemist, and some of you probably are, and many of you also are not, so just a little background here. The way we make ATP is one of the most bizarre features of all cells. That, I mean, you would never probably, you know, in your wildest dreams and, you know, the wildest drugs, you'd probably never think of this as a way to make cells. We use a thing called ATP synthase. It sits in the cell membrane, and so this would be the double membrane of a mitochondrion or a bacterial cell. And of course, we go through the electron transport chain the way this works, and we pump hydrogen ions out, and then there's a hydrogen ion gradient. The hydrogen ions come back through this ATP synthase, and this is like a little turbine. It's swinging around at a very, very high rate, and every 3 clicks in the ratchet, you get an ADP to an ATP change. This is obviously a strange way to make energy, but we all do it. We all make ATP this way. This would be like building a dam on a river and then, you know, failing to realize, well, there's no water in the river. So we have to pump the water up above the dam so we can convert the mechanical energy into electrical energy after it goes through the dam. We don't usually do things like that. I'm told the French actually do this at night when nobody's looking. This is what my French postdoc told me. So this is how we make energy, and bacteria do it. The important point here, this is called chemiosmosis. And the important point is bioenergetics are restricted to membranes of cells, and the only membranes in bacterial cells are the plasma membrane. So that's where this all goes on. And then this is simple geometry. The idea goes that as cells get larger and larger, you're running into a surface area to volume ratio problem. So how do you solve the problem? Well, you could solve the problem by not having, the argument goes, by not having ATP synthase on your cell membrane or leaving it there but supplementing it somehow. The way we do it, we actually relinquish the use of we being eukaryotes, we relinquish the use of ATP synthase in the cell membrane, and they're restricted to the inner membranes of mitochondria. So membrane bioenergetics is still used, but the location has changed entirely in the eukaryotic domain. And so if this hypothesis is correct, then there sure better be as much membrane area in mitochondria as there would be on the surface area of the cell. So this is the first thing we took a look at. This is the surface area. We're talking about eukaryotic cells now, and we're asking, what's the surface area of the mitochondria in cells of these different species, and how does it compare to the surface area of the cell? If it's a one-to-one relationship, would be that line, it'd be a complete wash. You wouldn't be gaining any membrane area by using the membrane. It's the same as you've got on the surface of the cell. Now, and you can see that most of these points most of them are below the one-to-one relationship. Now, this is the outer membrane of mitochondria, and you might argue, well, the ATP synthase sits in the inner membrane, so that's what we really should measure. But typically, the inner membrane is no more than about 5 times the outer membrane area, so this really doesn't get you very far. I mean, it maybe pushes you a little bit above the one-to-one relationship, but it's certainly not a quantum leap in membrane availability for bioenergetics. But it gets worse than that, because it turns out eukaryotic cells don't use all the membrane to put their ATP synthases. In fact, they all reside on these inner, you've all seen these iconic views of the mitochondria with all these invaginations. Well, the ATP synthases just sit on the ridges of the cristae inside mitochondria. So most of the membrane is not being used for the residence of ATP synthase. You can also look at this problem in the following way. For organisms, we have single-cell proteomics. It's possible to figure out how many ATP synthase complexes, and it is a complex, how many of those complexes sit in the average cell. And so then we're asking for all these, there's bacteria here, those are the 2 yeast, of course, and these are our vertebrate species. And we're asking, how does the number of ATP synthase molecules, the total number, average number in a cell, compare with the cell surface area? Now, again, in these guys, they're not living in the cell surface area, but what we want to know is the total number in the cell living in the mitochondrion comparable to what, if you were just to predict how many would be in the cell, if they were in the cell membrane, what would it be? And you can see this is a nice extension. So there's no sudden boost in the number of ATP synthases in eukaryotic cells. There's no higher number due to the fact that they get to populate the mitochondrion. And you can see the scaling is pretty close to 1 on a cell surface area basis. And this is just the number of ribosomes. This also can come from single cell biology. The number of ribosomes per cell is, in some sense, a measure of the activity of a cell in terms of making proteins and so on. And you can see there's no quantum leap in what goes on in eukaryotic cells. So we're seeing this repeatedly. There's generally this continuity of scaling that we see between what you see in bacteria and eukaryotic genomes. So it's sort of suggesting that really nothing, things changed, of course, in terms of cell architecture, but in terms of the overall bioenergetic efficiency, it's as though nothing really did change. Now, finally, I'll talk briefly about lipids, because this is another problem. Of course, mitochondria, they have dual membranes. Each is a lipid bilayer. Lipids are expensive. And so one can, from basic biochemistry, you can figure out how much it costs to build a lipid molecule. And this is sort of it's not important you understand where this came too important, but you can come up with a simple expression for the cost of building a lipid. It's a function of the number of carbon atoms in the fatty acid tail there and the number of double bonds or the degree of unsaturation. You can calculate this. And I've already pointed this out, that that's the cost of an average amino acid. It's about twice the cost of a nucleotide. This is the cost of, there's a wide variety of lipids that go into cells, but the cost is somewhere between, depending on the flavor, between 200 and 600 ATP. So lipids are quite expensive. So what we want to know is, if you're going to make a mitochondrion or an ER or a Golgi, whatever, a nuclear envelope, yeah, it's nice, it can do nice things for the cell, but how much are you paying for this intracellular infrastructure? And so we can calculate the price of mitochondrial membranes in the following way. The total cost of membranes in the mitochondrion is the number of lipid molecules per unit surface area in the mitochondrion. We know what this is because people have measured the head group area for lipids. And in fact, this basic idea goes back to an experiment done by Ben Franklin, I guess 200 or 300 years ago, whenever Ben Franklin existed. And Langmuir did similar work. You can figure out the surface, he did it without even knowing any detailed molecular biology. But now, by understanding the structure of lipids, we know how many molecules would be present per unit surface area. I just showed you the cost of building a lipid molecule, and then for a wide variety of bugs now of different cell volumes, we know the surface area, and there it is. It scales linearly with the volume of the cell. So this is the total mitochondrial surface area, inner plus outer membranes. And so we can take this and plug it into here. This is that equation I just showed you, and we know for a whole wide variety of organisms what lipids are used in the membrane, so we can calculate the average cost of all the lipids being put in membranes. This is the total cost of lipids. We divide that by the energetic cost of building the whole cell. So then that gives us the cost of making mitochondrial membranes, what fraction of the total energy budget of the cell it is. And this turns out to be, it almost is independent of cell volume. It's about 5% of the cell's energy budget. So eukaryotic cells, about 5% of all our energy is being used just to build those membranes of the mitochondria that we need to provide us with the energy to build everything else, including them, if that made any sense. So finally, I'll close with a few other observations on membranes, and this is where we're really lacking in information. We have good microscopy techniques like focused ion tomography and so on that now enable us to go through cells like eukaryotic cells and just shave layer after layer after layer, and then you can figure out how much is allocated for all the membranes of all types inside the cells. But this is all the data we have at this point. There are cells of very different volumes here. So this is Ostriococcus. This is the smallest eukaryote that you can grow in the lab. It's smaller than many bacteria. That's yeast. That's Dunaliella. That's another green alga, and that's cells from the pancreas of pigs. I guess there's a lot of pig pancreases lying around to do this kind of work. So what one can ask then is what fraction of the total energy budget of all these cells is just going into membranes? These are all eukaryotic cells. And what we find is about 10 to 30% of the total energetic budget of a eukaryotic cell is going into making membranes. So that's the sum of all the membranes, the ER, the Golgi, the nuclear envelope, the mitochondrial membranes, and so on. And of that cost going into membranes, about 30% is spent just on the mitochondria. So yeah, mitochondria are making energy for us, but we're paying a price just to build mitochondria. So it's a nice consortium that we have. So the closing point here is that it doesn't appear that the colonization of the mitochondrion really caused a bioenergetic revolution at all. If anything, it looks like we eukaryotes are somewhat less energetically efficient than bacteria. And the way it looks to me, if we want to be generous, it looks like the original consortium between the mitochondrion and the host cell is more or less a zero-sum game. We just look energetically as though we're just scaled up bacteria, although, as I said, we do things in very different ways. So I don't know what time it is. I'm pretty sure I haven't gone over time. So that's, pardon? Well, I'll just end here. This is the, there's a few people in here I don't seem to recognize, but this is my lab. Hopefully they're not my grad students. This is the guy who runs the lab, Dr. Tom Doak. We have money to do research, and I still find it hard to believe somebody's given me a job, and I'm very grateful for that. It's the only way to do this kind of thing. So I think that's all I wanted to say, and hopefully this will be of some interest to some people in the audience. Thanks.
Thank you, Mike, for this talk. Any questions? Yes, please.
Michael, the cost of transcription could explain the massive transcription for the genome that we guess it's totally useless. So this 80% of the genome that gets transcribed. So selection cannot act against this 80% and get down into maybe 5% of making things.
Yeah, that's a good point. I failed to make that point that, and as many of you know, there's been some entertaining debates about what's functional DNA and so on. There's a lot of DNA that just seems to be transcribed without any obvious function. That doesn't mean it doesn't have it, but the point here is that in big multicellular, even in metazoans like fly and worm, the price to pay for that is actually not all that high relative to the total energy budget of the cell. So it means there's a lot of slop in the system if just transiently you had an increase in transcription for some segment of DNA that has no function at all. As long as it's not toxic or something, selection would not be able to reduce that level of expression. That would not be true, though, in microbial species.
I have two comments, and interesting what your thoughts are. So you mentioned that there's this speed limit that you're not sure what causes this speed limit between volume of a cell. And so one obvious factor would be that in a cell, a significant volume of the cell is due to ribosomes, right? And so there is a certain maximum speed of producing basically amino acids, right? And that will be the limit, because you need to produce ribosomes themselves, right? And this will be the major constraints on the volume. So you need to produce an engine, in a sense, that drive your car. And so you cannot produce basically amino acids faster than possible with ribosomes in that volume.
Right, and you may recall I provided some scaling for the number of ribosomes in a cell somewhere back. I guess it was here. And you can see it scales a little bit sublinearly on a log scale, right? But keep in mind that, and you see more or less the same scaling. So here's the eucs and here's the procs. And yet in bacteria, keep in mind that there isn't an increase in cell division time with cell size. So, ribosomes presumably have something to do with it, but I think that still doesn't answer why we don't just have more ribosomes in the cell, for example.
Well, but for a speed limit, in a sense, you can have more ribosomes, but then it will increase volume of a cell. So that will be exactly the speed limit.
Yeah, well, I guess that's what I'm saying. It's not clear that that's limiting. I mean, that's a question.
It's easy to check, so
Yes,
because we know that speed of translation is limited. You cannot make it above certain
you need
speed.
more ribosomes
and so
to
that will be
produce
the
everything else.
just how it scales with the size.
In trying to understand whether a piece of DNA hangs around the cell, to what extent do you need to understand not only the effective population size of the host, but also the effective population size of the element? So I'm thinking about virus or phages, for example. Or do they just cancel out because they roughly scale with the population size of the host?
Oh, you're asking about how would this apply to a pathogen, for example, living inside, say, a eukaryote?
Well, say you have a selfish element that spreads, a virus, okay?
Yeah.
Presumably that'll also involve reinvasion. So if you just model whether a piece of DNA persists in the cell based on the effective population size of the host, you might be missing a stronger selective force in a way.
So it's not clear that, I mean, there's a lot going on with different kind of pathogens, so I don't think one can say that the effective size of a pathogen is directly proportional in any way to that of the host. It depends on transmission and so on. There is one very interesting set of examples, though. There's these viruses called Paladino viruses, spelled polydNA, and I think the folks who work on them call them Paladino viruses. These are viruses in parasitoid wasps. They have actually transferred their genome to the chromosomes of the wasp. So I think there you have a pretty strong, compelling case of a viral genome that must have the same effective size of the host, because it's in fact part of the host chromosome. These viruses are very interesting. Many of them, as you know, viruses have incredibly streamlined genomes, overlapping genes, and so on. Paladino viruses have introns in their genes. They have sometimes mobile elements, strong, high intergenic DNA. So they've taken on the complexion, sort of the genes in their host itself. This doesn't get at the energetic issue, directly, but you can see that even if you transfer a viral genome to a eukaryotic host, it can take on sort of the kind of attributes of the host genes itself.
For the mitochondrial story, have you looked at how the shell volume? Because you have to do this thing where you pump and make a gradient. If you had all of your synthases on the surface, it seems like the shell volume would be quite a bit larger than with a similar number of synthases in mitochondria, and thus creating the gradient.
The shell volume. The cell volume? Or?
No, just the volume between the inner and outer membrane, where you need to make this gradient, right? You have to pump the...
Yeah, it's not much different than the width of the inner and outer membrane in bacteria, I think. Maybe a nanometer difference or something, but...
Right, but in a large eukaryotic cell, if you were to put all of your ATP synthases on the plasma membrane, on the inner membrane, you'd have to generate the gradient on the...
Yeah, I mean, we can't do it anymore because we don't have two membranes out there, right?
Right, but yeah. I guess part of your argument was that there aren't more ATP synthases, right? Because the surface area argument, it's about the same.
Yeah.
But what I'm saying is generating the gradient itself, is it more cost effective to do it in the mitochondria than it would be
That's
to
a
do
good
it
question.
on the surface?
It's unclear, yeah.
Because
There are some bacteria, of course, with single membranes, and they still use ATP synthase out there. So one way of thinking about it is you have to have a gradient, of course, but one way of thinking about it is you're making the gradient by pumping stuff out, ensuring that there's more out there than inside. So yeah, this is something Rob Phillips and I are trying to work out now. There's a whole series of things that still need to be done in this area, but I'm liking this idea of trying to figure out the baseline cost of everything in a cell, ion channels, you name it. And I think this will bear on a lot of understanding of how things evolve in cells, I hope.
Yes, please.
As I'm understanding it, the energy production and usage of the cell basically is expanding as the effective population size is decreasing. So the selective strength is also weakening. But to what extent is there maybe a feedback between these two, that as the cell is growing, the genome is growing, of course, then maybe the effective population size is decreasing, and that's then reducing selection even further, which is allowing further expansion of energy usage and production and so on, and whether that feedback loop could potentially be measured or estimated in some way.
Yeah, I think I'm following. It was a long question or point.
Yeah, so basically,
Yeah, there are potentially feedbacks, positive feedbacks here. I'm not at this point really in the position of invoking what's driving the evolution of cell size, right? And there's very wide range of cell sizes in eukaryotes, even within single groups like diatoms, dinoflagellates. There can be several order of magnitude differences of cell size. But the question would be, in these narrow phylogenetic groups alone, if you had all these data, and we're far from having this level of data, would you see this pattern repeated over and over in different phylogenetic groups? That would be sort of the dream situation down the line.
So in eukaryotes, if the cost of just producing things is below the selection threshold, then you suggest that maybe the most important thing then is for the cell to avoid gain of function in these elements, and so that there could be monitoring systems for that. But those systems themselves, of course, then generate a cost of maintenance. But we do have like RNAi systems and things like that within the cell to sort of, it seems now, to suppress these background level things. Have you thought about sort of the evolution of monitoring systems, and are they more prevalent in eukaryotes than in prokaryotes, for instance?
Well, I would say they're, without question, to our knowledge, there would have to be a higher investment in eukaryotic cells, I think. It's not to say folks don't do it. But yeah, it's on the list. We've sort of jumped from the stuff on the cost of a gene, as you saw. That the stuff on lipids is just something we've very recently gotten into, so there's still a lot to do, and that would be one of them.